Thank you for joining us. I'm Amanda McGill-Johnson. I'm the Executive Director of the Nebraska Coalition for Life-Saving Cures, otherwise known more briefly as Nebraska Cures. Our mission is to promote, support, and advocate for health science research for the betterment of our health and also for our economic health. Today, I'm really excited to be bringing this panel that draws attention to health science research that's funded by the Department of Defense. I know when I think of the Department of Defense, scientific research isn't the thing that comes to the top of my mind. And so this is a really great opportunity to dig a little deeper and to learn about some of this cool research that's happening here in the state of Nebraska. We're joined today by 4 expert panelists who will share their work and also take a few questions from viewers and from myself. And so for those of you watching this live, please feel free to put questions in the chat and we'll try to address those in a timely fashion. And we'll go ahead and work through folks in alphabetical order. I'll introduce everyone before we get started with the presentations. We have Dr. Tomas Helikar, an assistant professor of biochemistry at UNL. We have Dr. Aaron Likens, who's from UNO in the bioengineering area. Dr. Josh Santarpia with the University of Nebraska's National Strategic Research Institute. He's the research director for chemical and biological programs and is also an associate professor at UNMC. We hear a lot from him during the pandemic as well. And then Dr. Alicia Schiller, the director of combat casualty medicine at UNMC. So thank you all for taking the time to join us for this Lunch and Learn today. And with that, Dr. Tomas Helikar, I'll go ahead and turn it over to you.
Thank you. I'm going to go ahead and share my screen. Can you see my screen? No, not yet.
Not yet.
How
there
about
we go.
now?
Yep, see it now.
Okay. All right. Thank you very much for having me here today, and thank you for a great introduction. As Amanda mentioned, I am in the University of Nebraska in Lincoln, especially in the Department of Biochemistry. And today I'll share with you some of the research that we've been doing in the context of NSRI and the Department of Defense. And in particular, I'll talk about the work that we're doing towards the development of a immune system digital twin to help with drug development for not just Department of Defense, but also beyond. So as Amanda mentioned, I also did not realize how much life sciences research is going on in the Department of Defense. And one of the projects that I have been involved with is related to radiological preparedness and radiation countermeasures that are obviously key to national security for reasons including detonation of nuclear weapons or radiological dispersal devices, even unintended radiological or nuclear accidents. And one of the diseases, one of the health issues that are relevant to for the Department of Defense is acute radiation syndrome, which happens when you're exposed to radiation. And the challenge with the acute radiation syndrome is that it's a multi-organ disease. It impacts reproductive organs, skin, bone marrow, GI tract, lungs, eyes, hair. And it really depends, the extent of the disease really depends on how much of the dose of radiation you have been exposed to. Early symptoms include nausea, vomiting, diarrhea, but later symptoms, and again, in the case of high dose exposure, it results in internal bleeding and ultimately organ degradation, and it can be deadly within weeks. The challenges that we have is that currently there are no radioprotectors. That means what the Department of Defense is interested in is, can we develop medicine drugs that a soldier could take before heading out to an area that might be exposed to radiation and work in that area with limited consequences to their health for as long as possible. One of the reasons why we don't have radioprotectors today is that in terms of the big pharma and the private sector, there is very little to no market for this application for these drugs. Complicating things is also, as I mentioned before, it is a very complex disease, and the FDA approval process is also more complicated. Here we have the ANIMA rule that applies to developing drugs for the acute radiation syndrome, which enables us to use animals to show the benefit or efficacy of these drugs, because it would not really be ethical or possible to have human clinical trials related to radiation. And another challenge that is not unique to the Department of Defense is that it costs about $1.2 to $4 billion to bring a drug to the market. And this is for the pharmaceutical industry average. And it takes over 10 years to develop these drugs. So with NSRI, so I'm one of 74 fellows, proud fellows for the NSRI. And as part of the NSRI, we are exposed to a lot of different expertise that is able to come together to solve complex problems and complex diseases. And one of the efforts that's been organized or developed here under the NSRI is the Nebraska Drug and Discovery and Development Pipeline that's headed by Drs. David Berkowitz from UNL and Dr. Ken Bales from UNMC. And essentially, the Nebraska Drug Discovery and Development Pipeline aims to become sort of a virtual pharmaceutical company that is able to go from, for any particular disease, in this case, we're focusing on acute radiation syndrome, and go from drug target discovery to preclinical studies and all the way to clinical phases, clinical trials, and then ultimately FDA approval. And the advantage here is that this pipeline leverages the diverse expertise that exists across the University of Nebraska system. And it happens so that across the entire University of Nebraska system, we have the expertise that usually works in isolation, but now through this pipeline that we can bring all this expertise together and we can implement essentially this drug development pipeline here in Nebraska. And so when we talk about the acute radiation syndrome, I mentioned that disease is very complex because it affects multiple organs. But if we look across other diseases, most of them are equally complex. This is still very fresh in our minds when we think about COVID-19. That disease is also extremely complex. It also impacts different cells in our body, different organs in our body. We all respond to the virus differently depending on our pre-existing conditions or health status and so forth. And not only are these diseases complex in that they impact different organs, but when we start diving deeper into our body, into the individual cells are also extremely complex. And so how do we tackle diseases, and how do we tackle this complexity of biological processes that govern our health, that are responsible for keeping us healthy? And so that's where our lab focuses a lot of our research on, is that we build computer models of the diseases. We build computer models of these different biological processes so that we can predict potentially diagnosis, prognosis, we can identify new drug targets, we can optimize therapies. And in most recently, over the last couple of years, we've been focused on building what we call a digital twin of the human immune system. So We want to be able to we're considering the data from the body, different organs, tissues, what happens across the different cells as they talk to each other in our body, but also what goes on inside of these cells. We build mathematical model to the point where we can build a digital twin of the human immune system as a system as a whole. And when we talk about digital twins, in manufacturing, digital twinning is a process that has been used for many, many decades. In particular, where we take an object, for example, it might be a jet engine or it might be a car, and we create a computer representation, a model, a computational mathematical model that represents that object. And that then enables us to optimize the model to teach us how we should optimize that real object. And so what it does in the long run is that it saves money and accelerates the research and development of these modeled objects. And in our case, we can use these mathematical models to develop, hopefully, to speed up, accelerate drug development, and also develop therapies that might be more personalized towards certain subpopulations, for example, or towards populations with different pre-existing conditions and so forth. And they can also help us in terms of the diseases where human clinical trials may not be as feasible, such as the acute radiation syndrome, where we can augment the use of animals for these studies with these computer models. Imagine you have a digital twin of yourself, but then digital twin that can simulate a virtual patient population of millions of virtual patients that we can then simulate the impact of a particular treatment on, and simulate many more conditions that you could actually be even able to capture in a real human clinical trial. And so our group, to be able to do this work, our group is very interdisciplinary. We have staff in my group that do software development, mathematical modeling and simulations, artificial intelligence and machine learning, high performance computing, as well as immunology and biology. And so, as I mentioned before, we have been applying these mathematical models now to the acute radiation syndrome that's been funded in our lab by NSRI and DOD, but we also have NIH funding that enabled us to actually identify, by modeling host pathogen interactions, we've been able to identify novel targets for potential antiviral treatments, and right now specialized to influenza, but ultimately we can expand these insights into other viruses. And so we're currently also doing preclinical studies, efficacy studies, and toxicity studies for our new antiviral drugs. We've also applied these computational models to identify novel drug targets for different autoimmune diseases. And as we expand our computational capabilities and the model grows to ultimately maybe one day be a digital twin of ourselves, we can also continue to expand on the diseases that we focus on. So with that, I want to thank you for your attention, and I'd be happy to take questions whenever the appropriate time is, when Amanda lets us.
No, that's great. If anybody does have any questions, please put them in the chat. So this really is an example of how there are blended funding streams that can help with more universal goals that aren't just maybe for the military or just for the general public. Okay. Well, I don't see any questions at this point, so we will go ahead and move on to Dr. Likens.
Great. Let me just get my screen sharing. Okay, can you see that okay?
Yes,
Good.
it's going.
Okay, good, great, thank you. My name is Dr. Aaron Mikins. I'm an assistant professor in the Department of Biomechanics at UNO. also the director of the Nonlinear Analysis Core, which is a service center that's part of the Center for Research in Human Movement Variability here in the Department of Biomechanics. I would like to thank the organizers for putting this together and giving us the chance to talk about our research. This particular project that I'll talk about today is relatively new and still very much ongoing. And it's really focused on understanding the effects of prolonged physical stress on the warfighter in terms of fatigue and changes to their movement patterns and some of the underlying physiology and so on. And so for the next few minutes, I'll sort of elaborate on the specific context of the research and talk about sort of the goals of the project and so on. So if you're not aware, US soldiers routinely engage in something that's called a Ruck March. And if you're not familiar with the term, which I suspect if you're on this military research symposium, then you may be familiar, but if not, this is a process where soldiers sort of put on these really heavy backpacks and all this equipment that weighs quite a bit. And then they go out and march over rough terrain. It's variable, and they do this for several hours. That can mean that they're carrying up to half of their body weight. And just to give that some perspective, if a soldier weighs 180 pounds, that's a lot like going to Lowe's or Home Depot, buying a giant bag of concrete and then carrying that with you for 3 hours while you hike around Fontanelle Forest or something. So it's pretty demanding, and it's not just a single day. Often they will engage in these activities for multiple days in a row. So it's a pretty impressive physical feat that they engage in, and certainly highlights, I think, their dedication and endurance. That said, I think it doesn't really stretch the imagination to sort of come up with a sense of how taxing these marches could be on a soldier's body. Like I hinted in the previous slide, these sort of extreme physical activities, these prolonged bouts of physical stress, are extremely demanding. How demanding? To give it some additional context, the Army Public Health Center recently noted that musculoskeletal injuries are the primary health concern for the U.S. military, especially those that result from accidents and chronic repeated stress, like they are sort of engaged in with these Ruck marches. In fact, they report that about 50% of U.S. soldiers will experience an injury related to overuse sprains, and stress fractures of their lower extremities that result from these kinds of activities. Right? So I think even just based on those numbers, you can get kind of a good appreciation for how profoundly these activities can affect their movements and, again, their underlying health. So the goals of this project are sort of multifaceted. One, we don't really know yet enough about how these kinds of extreme physical activities actually do affect soldiers' movements and their underlying physiological consequences. So one of the goals is to better understand that by studying their activities or their movements and so on while they're engaged in these activities. The other sort of goal is to actually be able to predict those consequences before they happen by developing software and a toolkit that can hopefully monitor soldiers as they're engaged in that. I'll talk about that more a little bit. And then, of course, once they do experience this sort of intense fatigue, it would be good if these tools that we're developing could actually provide some insight about when a soldier is ready to return to full active duty. So the context for this work is a collaboration between myself and some researchers at the U.S. Army Combat Capabilities Development Command Soldier Center, which is quite a mouthful. So usually I just say CCDCSC or Soldier Center, but that's the group that's collaborating. And this particular larger study is a laboratory-based simulation of these Ruck marches. It's a multi-day Ruck march study. So on the first day of the study, the soldiers arrive and they go through a series of physical and cognitive and emotional assessments and so on. And then over the next few days, they perform a series of these Ruck marches that are interspersed with assessments of fatigue and marksmanship and other activities that soldiers sort of routinely engage in. And during that procedure, we're measuring dozens of variables at really high sampling rates. And we're talking about 100 or more times per second. And so over these several days, the amount of data that we're collecting is just absolutely huge for this kind of activity. And then finally, on over the last few days of the study, after the Ruck Marches have been completed, they're monitored for recovery to see when they sort of return to their baseline levels. I would love at this point to share with you some nice data that we've collected about these exciting results, but unfortunately, the data collection has just actually ended, and we're still in the process of processing the data and transferring all this stuff from the Soldier Center to UNO. But I can talk more about the sort of goals of the project and where we are in going forward. Oops, went backwards, sorry. Okay. So the sort of final goal of this project is to develop what I call the Readiness Toolkit. Essentially, this is a software suite that analyzes various aspects of human movements and physiology. And on this slide, I actually got a small glimpse of sort of what this could look like. So the yellow trace, you've got essentially 10 beats of a human heart. The blue trace here are actually the lower leg moving in the sagittal plane, and then there's several sort of respiratory cycles measured by their respiratory flow. And the thing that I'd like to appreciate from this slide is the variability in different biorhythms and signals that we have to collect and how different they can be. Sometimes they're very smooth and regular, and sometimes they can be quite complex, like you see in these cases. And that complexity really only sort of explodes when the task is to understand how all of these various biorhythms coordinate in the service of producing these movements, even simple movements, but certainly in these extreme physical activities, and then learning how those coordinations sort of break down when we become fatigued and can no longer perform our movements. Obviously, that's certainly of concern. So the Readiness Toolkit is just a set of, not just a set, but is essentially a set of cutting edge analysis techniques and methods that are really naturally suited to studying how changes in movement and physiology can result from injury and disease. And we're applying this to this particular problem involving prolonged physical stress in soldiers. So all of these methods that I'm sort of talking about in the previous slide will ultimately provide us with this huge, really rich feature set that we can use to model soldier fatigue over the time course of this multi-day scenario. Now, where we want to go from there is that we're going to use various machine learning tools to select the absolute best set of features that we can use to model soldier fatigue. From there, we will move into the next phase where we're going to develop really efficient algorithms based on what we've learned that can be deployed essentially in real time. For example, using lightweight wearable sensors that can actually detect these kind of changes in their movements and their physiology, hopefully before they actually do experience injury or disease. That's sort of the end result of this goal. We're just now sort of entering into this phase of the research, but definitely very excited about continuing to work on this over the next couple of years. So just to summarize, this project is really focused on prolonged bouts of physical stress and how they affect soldiers' movement and their physiology. Developing this software based on my expertise in time series analysis and also movement science, human movement science, so that we can identify sort of the key markers of soldier fatigue and their underlying movement patterns and its variability. I think that the prevalence of the stress-related injuries that I talked about earlier, I think really highlight the timeliness for this kind of research, and really I think underscores the potential to sort of directly benefit the men and women that are fighting to protect our country and risking their lives on a daily basis. And with that, I'd like to thank the National Strategic Research Institute, this really nice, unique sort of resource for the state of Nebraska that provides this great mechanism to obtain funding to do this kind of work, and also my collaborators at the Soldier Center for working with me on this project. So with that, I'll thank the rest of you for your valuable time and attention, and I'll take any questions if there are any.
This is really great. Just, I know a number of veterans who've come out with chronic injuries or some level of disability due to just those, like you said, ruck marches and all the weight they're carrying. And so, just maybe reiterate or ask a follow-up question. So it's about identifying kind of those moments where it's starting to hit that tipping point of potential longer-term injury and trying to hopefully there could be some intervention then at that point to
right. So, ultimately, if we're successful, that would allow sort of real-time or on-demand type monitoring of their activity, and could provide their commanding officers with information to help better, guide decision-making about what they should and shouldn't be doing at that moment.
Great. And I'm assuming there'll also be some just realizations around how to help people come back from it too, who've maybe hit that tipping point, and how do you help them appropriately recover, gauge that time period.
Absolutely. And what is that appropriate time period, and so on. Yeah, absolutely.
Yes, absolutely. We have a nice comment from Bellevue City Councilman Don Preister about appreciating the study info. Also like to work on preventing war, so that we're not sending people into harm's way, of course, would be an ideal situation. But I'm certainly glad that work is being done to help our soldiers not get injuries that aren't maybe absolutely necessary in their line of work. So thank you. I don't see any other questions. And so we will go ahead and move to our presentation from Dr. Santarpia. Here we go.
On mute. Sorry about that.
Oh, there we go.
See. You guys seeing the slideshow now?
Yes, we can see it.
Very good. So so thank you for the introduction. Dr. Josh Santarpia. I am in the pathology and microbiology department at the University of Nebraska Medical Center, and I'm also the research director for chemical and biological programs at the NSRI. And I'm going to talk a little bit today about the intersection between the COVID-19 research that was done at UNMC and NSRI, and how that intersected with the military work that was like sort of all of the intersection points between this sort of pandemic research that we were doing and military national security programs. So it's actually a relatively complex space. I'm going to talk about four particular efforts, sort of some of the work that we did initially to characterize human-generated SARS-CoV-2 particles, some of the work that informed the need for understanding some of our transport systems and military transport, and then some of the work that we're currently doing for Department of Homeland Security, looking at hazard characterization and protecting their workforce, CBP and TSA folks. All of these programs and the others that I might mention in brief are sort of blended collaboration between NSRI researchers and UNMC and University of Nebraska researchers. So some of the first things that came out during the pandemic, of course, were concerns around how the disease was transmitted. Upon receiving our first cohort of patients that came from the Diamond Princess cruise ship, UNMC was the place where our government decided that they should be cared for and housed until we had an understanding of, a better understanding. And so we had several people in quarantine, and we started doing some study around those folks who were positive with COVID-19. And what we found was very concerning, of course, that there was a high degree of contamination. Most surfaces in a person's room at that time were contaminated. We had evidence of viral RNA and aerosol particles and in air handling grates in the rooms. And, there was some limited cell culture that seemed to indicate that some of these might be capable of reproduction, and so they would be actually a hazard. And we also noticed that we didn't have direct correlation to symptoms and this environmental contamination, meaning that someone could be contaminating their environment or be shedding lots of virus into their environment without really displaying significant symptoms or symptoms that we might, some things like body temperature that we might pick up by body temperature scanning people. So if you kind of remember, this was back in February and March of 2020, which seems like a completely different world now. And so we were really just beginning to learn about this disease, and some of this first work was done here. Later in the, a few months later, we were trying to determine really more specifically what size of aerosol particles contain not just viral RNA, but also infectious virus. And we were able to determine that it was in fact quite small particles, maybe particles less than a micron, that contained infectious virus, which is consistent with those that would be produced in the deep lung. And knowing that this is, some of the initial results of this, Tom did a good job of describing the vast variety of tissues that SARS-CoV-2 can infect and how it might differ in presentation from person to person, but knowing that the pneumonic form of the illness seems to be fairly common. Knowing that, like, the expectation that very small particles that would be generated in the lung might carry infectious virus was something that we were pretty concerned about, and turned out largely to be true. Other studies have also now shown similar results, citing very small particles containing infectious virus. So at about the same time, and this is sort of the military intersection part, at about the same time, we were getting asked by our Air Force, the Air Mobility Command, about moving people infected with COVID-19. We at UNMC, I think it was a UNMC, UNO, and UNL collaboration, had done some work evaluating a military transport system called the Transport Isolation System, or the TIS, several years ago around moving Ebola patients. And they came back to us and wanted a better understanding of how they could adapt that system to deal with an airborne disease, as we were beginning to understand that this was likely an airborne disease. And the military's position on this was that they were going to handle it as if it were, regardless of what the prevailing thought was. That was the best case scenario. So we worked with members of the Air Force and a huge, actually, interdisciplinary team, not only to rework protocols for AMC to help use the TIS, because that was the system they had in hand, but also to evaluate and develop another system called the PBCM, which is just a hard-sided transport system. And these are all flyable systems, right? So this is the kind of thing you would load into the back of a large transport aircraft, bring infected people in, and you can provide both care in the air and transport back to the United States for hospital care. And so the TIS system, which we had helped them validate several years ago, was actually safely used to fly many missions with COVID-19 patients back to the United States. It's largely military members, repatriating them from wherever they were. We helped work with them to develop their own hard-sided system to transport people who needed less care and more, who were still positive and needed isolation, but not necessarily care all the time. So that was sort of where it started for the intersection with the military. Many of you have probably heard a lot about the work that my group had done with Transcom and looking at civilian aircraft, which is actually on the next slide, but we also did a lot of military aircraft, which we were looking to transport large numbers of troops where someone may or may not be positive for COVID-19 at the time. And so we did aerosol transport systems to try and characterize how aerosols that would simulate COVID-19 particles would move around the aircraft, both during flight and on the ground, to try and give an understanding of what the risk would be for any given airframe that might be used to move troops during the period, and what risk they were putting their pilots at, their care staff at, and other potential passengers. We also looked at civilian aircraft, which is what got, I think, the most press attention in the last year. And we looked at 777 and 767 data, or 767, and we did these same kinds of tests. And there's some images at the bottom here that kind of look at the kinds of experiments we set up on this with the mannequin head that was producing particles that we could mask or not mask, and detection systems that were placed throughout the aircraft. And this was really to try and understand the risks and hazards. Wasn't initially meant to be applied writ large to air travel, but really to support the movement of Patriot Express flights and moving troops, again, moving people back, repatriating soldiers into the United States, where where we were concerned that someone might have COVID-19, not necessarily that we were moving infected patients. So it really supports both. So in summary, a lot of the airframe testing that we did demonstrated a significant variability in the risk, depending on what aircraft you picked or what ConOps you use, or whether or not people were known to be sick or not known to be sick. But the military was able to use that data to help inform risk and plan troop movements during the pandemic, which is important both to providing healthcare for these people, but also just for repatriating people as the world sort of closed down in 2020. The commercial airframe testing helped inform risk for these Patriot Express flights. And I put in a few things here just for everyone to see from an aerosol perspective. Exposure risk in flight is actually quite low. If your risk of exposure to someone else's aerosols, even if they're sitting quite close to you, is actually quite low. However, there are lots of variables that come into play when you add human movement. It isn't just an aerosol generator or someone facing forward. And so you have to sort of consider the other factors when you think about, what it is like to be in close quarters. But in terms of the built environment and a space where you would think that with lots of people packed together, your transmission risk might be significant. And some very, that environment is actually very, it's more protective than you would think a giant flying aluminum tube would be. So it speaks a lot to aircraft ventilation systems and how far that's come. Now, I think there's, like I said, there's a lot of other factors with regard to travel that create risk, not the least of which just simply taking off your mask and eating, or, getting up and moving around, and the impact that has on the airflow can change things. In addition, everything you have to do to get to the airplane, walking down the jet bridge, which is not the airplane, and as knowing if everyone has flown to some degree or the other. So the idea that the airplane is always on and operating at full capacity, I don't know how many times I've sat on a hot tarmac with a plane turned off. It really, you don't have the benefit of that built system in those environments. And so all those things have to be considered, and were considered, in fact, when developing and designing these Patriot Express flights on these aircraft. So it was, that was sort of that intersection. As we go back to, sort of the present day, we're doing a lot of work, not directly for the military, but I think this informs a lot of operations where you have people who are interacting with a variety of people over time. And we've been doing sort of ongoing assessments of viral shedding into the environment of hospitalized patients, looking at PPE use and whether or not there's aerosol generation or risky, what the risk is, like donning and doffing PPE, looking at testing looking at environmental contamination in testing centers where we have known populations of COVID-19 positives, we get that data. And then looking retrospectively at how things are changing with respect to the variants and over time. And I will say a little bit that as we move through this sort of these waves of variants and the concern around them, the interesting thing about the current wave, the Delta variant, is that there does appear to be more likely to find the environment contaminated around someone who has a Delta variant than in any other time during the pandemic. And I believe that's likely due to changes in the viral titer. So people with Delta are producing more virus for longer periods of time. And so I think we're seeing them now in the hospital with still with very high viral titers, where before, a lot of times, by the time someone was sick enough to come to the hospital, their viral titer was actually starting to drop as their immune system took over, and they were not always the case, but I think this is sort of the trend that we're starting to see, is that more virus for longer periods means that we see them more in the hospital, and that someone with a Delta variant infection is more likely to significantly contaminate their environment, including an aerosol. Some of the other work that we're currently doing, and I'm going to try and wrap this up because I'm probably running long, looking at intrinsic factors that impact stability of viral aerosols, trying to understand what parts of the virus make it more stable, make things that are respiratory transmitted capable of doing that. We're looking at coronaviruses and hantaviruses as a model. And then the other things that we've done that have, maybe not that impact sort of the public good, I guess here, for those of you who like vacations, we've done work on cruise ships, similar to what we did on in the aircraft. We've done looking at public schools and transmission in public schools, transmission in the meatpacking industry, which will sort of be coming around to publication soon. We've looked at a number of novel antiviral materials, and we're working now on a DARPA program to revolutionize PPE and how PPE is used by probably military first, but everyone. So that's good for me.
I have a quick question.
Sure.
I feel like early on, there was a lot of talk about transmission within the military or some different Navy ships, things like that, as kind of test grounds early on. I haven't heard much since then. And so do you feel like they've done a pretty good job of taking what they've learned, in terms of troop movement and such, and activated it in such a way that they're spread to a certain extent?
Yeah, I really think that there was, you can, having been involved in all of the discussion around the airframe work, and when they were talking about troop movement and things, and there was just an extraordinary amount of hurry up. We were, they were, like, we're not flying a mission until we know what's gonna, until we have an idea of what's going on. And so we were, it went from 0 to fully functional operational field testing in like 4 weeks or less. I don't know. It was very rapid and very stressful, but it was, yeah, there was a lot going, especially because we were dealing with a pandemic at the same time. So it was very, they were very aggressively trying to do the right thing. I think the Navy ship thing that you mentioned, like, they were, everyone talks about ships having poor environmental conditions. And I think that, the age of the ship has a lot to do with that, and how, what was done with in that particular environment, how its airflow is structured and things like that. And I think that applies to a variety of things, including like cruise ships, where I think there's a pretty big spectrum of HVAC design and how that impacts risk of infection.
So do you think they were able to activate and really just all in right away because they had a funding source, like a strong federal funding source, much like with vaccine development, that they could say, this is a priority, we need to move our troops. So that federal funding piece was just really critical.
Yeah, I think, not just this, but just the entire United States response to the pandemic demonstrates what you can do when someone's just willing to write you a blank check.
Yeah.
Not worry about the money and just worry about the results.
Mm-hmm. And then I'm sure what they discovered obviously helped inform other decisions and other research, as you went on to other types of aircraft.
And I hope that it continues to inform the way that, over the next, decades, that what we've learned in the last 2 years helps to inform the way we do things. Changes maybe the way we do things.
Absolutely. Well, okay, we will move on to Dr. Schiller.
All right. Hello, everyone. Let me share my screen here. All right, so I'd also like to thank the organizers for the opportunity to present, and also everybody in the audience for listening today. My name is Dr. Alicia Schiller, and I am assistant professor in the Department of Anesthesiology and also an NSRI fellow, and have the pleasure of being our director of combat casualty care medicine on campus. Sorry. So what I kind of wanted to start with here was a little bit of a story, I think, of somewhat of an unlikely collaboration that I think has turned out really fantastically and opened up a lot of great avenues, I think, for UNMC as well as some of our military collaborators. So I am actually a physiologist by trade. So my area of expertise is cardiovascular disease, hypertension, hemorrhage. I worked in the DoD system doing device development and wearables for hemorrhage detection. So I was working with a chief of physiology doing such work at Tripler Army Medical Center in Honolulu. And what became really interesting was while we're doing all this hemorrhage work, you know, kind of being immersed in that military environment and interacting quite a bit with the military medics who were interested in what we were doing, we started seeing some kind of common things coming up with many of the medical procedures that they were being asked to train for. So that was really where this collaboration got started. And it just so happened that I'm also in the Department of Anesthesiology and have some collaborators in Department of Surgery that really have that expertise in those medical procedures. So that was really how this got started, which again, just kind of an interesting start and an unlikely collaboration here. So what I wanted to start with telling everybody about is intraosseous line placement. And we refer to this as a low frequency or high value life-saving procedure. So what that means is if you're going to perform intraosseous line placement, you're going to do it not very often. And when you do it, it has the potential to save somebody's life. So you also don't get to practice it very often. And what that's used for is if you need vascular access in emergency situations. So if you have lost so much blood that your venous system is collapsed and you can't get an IV in, the only other option you really have to do is an intraosseous line. And quite literally, you are using a drill and a needle, and you are drilling right into the bone, into this hollow cavity in order to administer fluids. Amanda, I see your face.
Sounds very unpleasant.
Yeah, so actually some of the ways this has traditionally been trained is the medics perform this on themselves. So they actually practice on their friends, and we had the opportunity to kind of sit in and watch this. So something that someone who's not a physiologist may not know, you actually don't have nerves in your bone. So you only feel the pain when the needle is going through your skin. And then also after you start infusing fluids and you have kind of pressure in there. So it's not as bad as it looks, but I certainly was not volunteering for this anytime soon. So one of the things in teaching this, right, and teaching a low frequency, high value procedure, medical simulation becomes incredibly important here, right? Because you're not doing it very often, but when you do, you need to perform this correctly. So one of the things we really noticed and we heard from the medics in discussing this was the anatomical accuracy of the models they had available to train were really poor. So I just have some examples for you here. Quite literally a bone with a patch put on it is, and this is what's actually provided by the company that is utilized that provides the needles and the drills. So I think you could see, of course, you can drill into the bone there and hit the right spot, right? That's pretty easy. And then as another example, we have kind of this package here, which contains another intraosseous line trainer. And you also see like there's these removable patches. So you really know where to put the needle, right? And that is actually the critical part of doing the intraosseous placement. So if you don't get it in the right place in the bone, you're not going to hit the hollow area. You could damage tendons or other parts of the patient's leg. And when you use these trainers that have these removable patches, these pieces are also really expensive. And we don't like to talk about that a lot, right? Because I think, there's a high value in medical simulation, but also if you're performing a task that takes a psychomotor skill to learn, you need to do it multiple times. And if you can't do that, you can cognitively understand how to do the task. But when you're really pressed for time, like you're in the dirt, your friend is screaming and you need to put an intraosseous line in him, you may not really be able to execute that task very well. So we also noticed that, with this collaboration that we had and sort of serendipitously, we were sort of having these issues in the anesthesiology departments with the physicians also trying to train the residents on the exact same things, right? As they were not really happy with the level of simulation that they had access to. So, we have this problem with both academic physicians and also just military personnel. I also want to note too, like this kit here is listed for $866. And each replacement patch is $125. So for example, if you were going to do a set of repetitions, like, I don't know, practice a skill that would save somebody's life 10 times, that's going to cost you upwards of a couple $1,000s in just the replaceable patches. So if you multiply that by 30 medics, you've really upped the cost here to learn this procedure. So again, serendipitously, at the same time, there was a attending in the Department of Anesthesiology, who I know also works with Dr. Santarpia. So Nick Markin had done some work making high fidelity scan-based trainers. And what that means and how that process worked is he was taking actual data from a CT scan of a person, right? So that's important because it's real anatomy. It's not kind of like sort of simulated anatomy, it's from a real person. And through some fancy computer work and some 3D modeling, it's actually able to print out the internal structures of the bones from these CT scans, and then also reverse those images and make a mold so that he could print kind of the reverse of the flesh, right? And then we put these together, place the bone inside, and then we're filling them with a sort of gel substance that we could actually break down and reuse. And we end up with a test trainer that has completely accurate human anatomy and is also reusable and reclaimable. So for us to make one of these models, just in terms of supplies, cost us about $7. Right. So, and again, you can reclaim them and remake them. So you don't necessarily, you could reuse the models without having these kind of training scars of being able to see exactly where you're trying to place that interosseous line. And then I just sort of wanted to show you a little bit more close up on what these look like. So for this panel here, we have a leg. Here's kind of our knee area and then also our shoulder area. So the two common places where interosseous line access is done. And then we also have some of the models that are made with a more transparent material. So on purpose, you can actually see the bone structure through them. So just to kind of show you how that's placed. And this can also be used as a training modality because you can palpate on this material and then also feel the underlying structures underneath. And then this is sort of what it looks like in practice. So if we do a simulation for the residents, et cetera, we drape them up, we give them the real tools. And then, if you perform interosseous line placement correctly, you should be able to aspirate bone marrow, right? So that indicates that you're in that cavity on the inside of the bone. And this is also incredibly important, and it doesn't seem that important, but I don't think there are any commercially available models where you can actually aspirate bone marrow. And that seems like a silly thing, but when we were having these conversations with the military personnel, they would tell us, well, I did this and, I used the drill and it went in, but you don't actually know if you did the procedure correctly. So you don't have that feedback at all. So something very simple can actually result in a training period where you're training yourself to do something wrong because you've never gotten the feedback to know if it is correct. So because these models are reusable and we're 3D printing them, we're able to replicate that hollow structure inside of the bone so they can actually pull bone marrow if they have been successful. So this project, again, kind of budding and has gone off in a couple of different areas, but we have been fortunate enough to be supported by the Anti-Tactic program. So that is between UNMC and Triplet Army Medical Center. We also have a task order that is supported through NSRI. And it's also resulted in a CRADA, which is a Cooperative Research and Development Agreement between the University of Nebraska and also Triplet Army Medical Center, which actually provides a significant amount of in-kind support for personnel that is at Triplet Army, and also to be able to utilize that military population for some of these studies. I'm breaking all the slide rules here, and I realize that, but I wanted to show you some of the data that we've been able to acquire kind of at this early stage. And this was presented at the MHSRS conference that was canceled. So I'm sorry, it was accepted. It was not presented. But what we did is, while offering training for intraosseous line placement, we were also able to interview a lot of the military participants and kind of ask them what they thought about the trainer, how that worked, how that was in comparison to what they were currently using, and also if this 3D scan-based model was even worth that time. And what was very interesting to us is the feedback that we received from the military population versus the academic population was very different. So the things that the medics were finding important and they really cared about were things that the academic physicians didn't care about. So one of the things that we don't really think about is just being able to have access to a task trainer, right? So because our task trainers are incredibly inexpensive, if one of them should happen to walk off, it's really not that big of a deal, right? However, if you have simulation equipment that's incredibly expensive, usually there are scheduling issues to be able to access it. It may not be in the same building. And those things seem nominal, but particularly in a military population where all of their time is accounted for, just having access to the simulation for as much time as they needed was actually a really large concern for them. So some of the other things that we learned kind of from this initial project, again, kind of looking between military and academic populations, is the way that we were defining success for when a line was placed between military and academic populations was completely different. Or should be completely different, excuse me. So again, the things that were important on a military side and how success of simulation on how we're judging them on the academic side should maybe not necessarily be the same from an educational standpoint. So for example, if you're an academic physician and you're putting an intraosseous line in the OR, it's really important that you make sure you sterilize that site, you put the appropriate dressing on, you do all of these things. That is absolutely not that important if you are in a military setting and you're in the dirt and quite literally you have to cut your buddy's pants off to be able to put a line in, right? So that was also something that was kind of brought to our attention. And we were asked to kind of help develop a curriculum that was a little bit more well suited to the scenario and also help develop training tools that were more suited for this specific military environment. Just kind of to wrap up on where this is now, we do have continued engagement with Tripler, and they actually visited campus just a couple of months ago to kind of discuss additional projects to see our simulation center. Our task order is kind of ending, so we're in that phase which all of us scientists are familiar with, where you have piles of data and nothing to do but write papers. So hopefully we'll have a lot of that published soon. And we were also asked then, because we felt like Tripler especially thought we found so much valuable information in just how their training was being conducted and how the actual personnel felt about that training. They asked us as well to conduct a needs assessment between the trainers and the trainees, basically, to see if their objectives are aligning with their training program for intraosseous line placement and also for several other medical procedures such as chest tube placement, which are really important for combat casualty care. So that is also in process. And I would be happy to take any questions, and my email address is also listed. Please feel free to reach out if we don't get to them today. So thank you.
Yeah, how cool is 3D printing and revolutionary in terms of being applicable to situations like this?
For sure.
We are getting close to finishing our hour, but I do want to, unless I see other questions in the chat, there was one I missed earlier in regard to Dr. Likens' presentation from someone in the audience. She asked, does the military flak jacket provide any back support during the Ruck marches? If not, would some type of lightweight back support help prevent some of the injuries and prolonged fatigue?
Yeah, it's a nice idea. Certainly you could develop some sort of brace or orthosis to help with the back support. But of course, anything else that you add is also going to add additional weight. So it's sort of a kind of a complicated issue. And it, depending on how it redistributes the mass, it may not help with the lower limb injuries as well, but certainly could help.
Okay, interesting. All right, but since we are at the end of our hour, I'm going to go ahead and just thank our panelists for participating and for folks who were here participating live and asking questions. Other than that, thank you for joining us and have a great rest of your day.